Thank you very much, Prash. And I love this idea of being an opportunistic extrovert. I would say that to everyone, that putting yourself out there, especially in the context of open source, being ready and willing to contribute to communities and to get involved with communities. I know that's what you've done, Prash, with bioinformatics.org, and that's what I did. It's simply being a member of bioinformatics.org, contributing to the mailing list of bioinformatics.org, trying to help people out. These things will get you noticed. And the brilliant thing about open source is it's totally a meritocracy. It doesn't matter where you are, what your age, nothing about you matters. But if you're willing to get involved and you're providing useful contributions, don't worry if they're not useful at first, but you can build on this reputation. You can build up a reputation in an open source community as someone valuable. And that opened doors with bioinformatics.org. That definitely opened doors. What Prash is doing now, for example, he said, I will proofread your cover letter. So take that opportunity. That's a really massive opportunity that you should seize. Definitely don't be worried about being opportunistically extroverted. If Prash doesn't review your cover letter in a timely way, pester him, because those are the kind of opportunistic, opportunities that you can seize hold of. And Prash, I would say, I'm happy. I love that idea. I love what you're doing with Bioclues, and I'd be happy to help proofread application letters and so on. I think that's a wonderful opportunity that you're offering people. So thank you very much. I was going to say I could go on about this topic for hours. I think really what we did with the nucleic acids research paper, again, it was about just getting involved with the organization there. Now, what was that called? The forum, SecWiki. It was a forum for next generation sequencing analysis. SecWiki was a great community. I don't know how active it is now. I built a structured database from the SecWiki forum in a way that anyone could contribute to, sort of open source philosophy. And for example, I didn't bring these people together across 5 continents. They came and contributed. They nominated themselves and they built on the database I had created because it was open source and anyone could contribute. So through that process, they selected themselves and they became the authors list. So it was really a phenomenal experience. And I would never have, if I hadn't reached the owner of SecWiki, if I hadn't just built that resource, it never could have happened. So just put yourself out there. I would definitely agree with that sentiment. So I was building SecWiki during my postdoc, but after my postdoc, I took a more service role in Ensemble plants. So I'll tell you now about this work I did at Ensemble plants. If I can make this full screen. Can you see and hear me okay? Great. So there's going to be a full screen here somewhere. I'll just press this. There we go. So I don't know if there's a hand raise opportunity. If you can raise your hand if there are any questions, I'd be happy to take questions through the talk. If anything's unclear, just shout and I will. I'd be happy to be interrupted to answer questions. So yeah, I was at Ensemble plants. I've repurposed this talk because I think it could be very useful. It sort of showcases some of the open source things you can do with Ensemble plants. It's a completely open data, completely open source code base, and you can use the services there. I was Ensemble plants project leader from about I guess it was a few years ago now, about 4 years ago now. I was there for 7 years. And I'll tell you a little bit about what we did, what Ensemble plants was in today's talk. So the motivation for Ensemble plants, which is a database of plant genomes and related data. I'll get into the data in a moment. Really, one of the main motivations for what we were doing was food security. There's a huge challenge in the area of food security, and I'll tell you a little bit about the challenge and our response. But I'll talk about something, I'll give you a couple of examples, what we might call the gene revolution. If you've heard of the green revolution, I'll talk about that briefly. The green revolution was really critical historically to meet the need of the food security for the globe. The green revolution massively improved crop production globally. And I'll talk about a couple of examples, and then I'll go through the data that we have today as sort of an overview. Now, as I said, this is a bit dated. I left Ensemble plants, let's say 2018, end of 2018. So if people are interested, I can send you an updated slide with the latest data about Ensemble plants. So this is just a slide about food security. This is world population growth for the last 10,000 years. So there's a logarithmic scale here of number of people on Earth. So this kind of always surprises me. Just 5,000 years ago, just not that long ago, 5,000 years ago, there were less than 10 million people on Earth. A very, very sparsely populated planet. And over time, we've seen this on a log scale, a sort of exponential growth in the number of human beings, along with, it's been facilitated by technology. So as technology has come along, it's allowed the population to grow. An important, technology has been agriculture and irrigation. I would add in here, I don't know when it first arose, but of course, sewage systems is an important technology for society. And you see here this sort of double exponential growth towards the 20th century. One of the massive drivers or facilitators of this growth has been fertilizer. So again, back to the agricultural innovation that's allowed this population to thrive. And fertilizer is very much related to oil and gas, as we'll see in a second. And about this time, if we look here, this is what we call the Green Revolution towards, I think, the 1950s. This Green Revolution was essential to support this population growth. I'll talk about that in a second. So if you look at just in case this panics you somewhat, I guess this isn't surprising for most people, but the WHO, the World Health Organization, projects population to actually stabilize. So we're not going to see a continuous exponential growth like a bacterial plate, but because of increasing development, population will naturally stabilize, or maybe 12 billion. So we're still facing a massive challenge. The population will stabilize. It's predicted to stabilize, but there's a huge challenge of food security currently. So this is a project I was involved with, the Wheat Improvement Program, Wheat Improvement Strategic Program. These are some of the press releases they've made. In the next 50 years, we will need to harvest as much wheat as has been produced since the beginning of agriculture. And you can see what that is with these double exponential growths. There's a huge amount of food required. This is from the Rice 3000 Genomes project. By 2030, rice production must increase by at least 50% to keep pace with global growth. And as a consequence, agricultural production will need to increase sustainably. It's an important word, with limited natural resources. So the UN estimates that by 2030, the world will need 30% more fresh water and 50% more energy. By 2050, we'll need 70% more food. That's a huge issue. So just to come back to this concept of oil and gas, this is a fertilizer production factory. So I was gobsmacked when I heard this statistic. I was amazed by this statistic. In your body right now, 80% of the nitrogen in your body has gone through the Haber-Bosch process. So this factory is using the Haber-Bosch process to create ammonia and ammonium-based fertilizers. Incredibly energy hungry. And it just illustrates how industrialized global food production is. That 80% of the nitrogen, so if you think about your DNA, the nitrogen in your DNA has been through a facility like this. Not all of it, of course, but most of it has. Let's say you have a very strict organic diet, perhaps you have less. But for most people eating most food, they're using fertilizers that have been produced in factories like this. So these factories use 3 to 5% of the world's natural gas. So if you think Bitcoin uses a lot of energy, global food production uses a heck of a lot more. So just coming back to the Green Revolution, this was really led by someone called Norman Borlaug. So he's credited Norman Borlaug, who promoted the Green Revolution through essentially two activities: increased use of fertilizer and more wide development of dwarfed varieties. So dwarfed wheat is massively more higher yielding than normal wheat. So the old wheats, the wheats of the 1950s, 30s and 50s were about 7 feet tall. So if you walk through a wheat field, you'd be completely hidden. Modern wheat is just about 2 feet tall, and that's reduced wind damage, and it's massively increased yield because the plant is putting more energy into the grain than it was now than into growing high, growing tall, because the wheat in the farmer's field doesn't have to grow tall. So in terms of efficiency and productivity, these dwarfed varieties that Norman Borlaug really led the development of have saved, credited with saving 1 billion lives. So Norman Borlaug personally is credited with saving over 1 billion people from starvation. He got the Nobel Peace Prize in the 1970s. And he said something interesting. I think I've skipped the slide. He said something interesting about how there's this sort of environmentalists in the West. He said if they lived just 1 month amongst the misery and deprivation of the developing world as I have for 50 years, they'd be crying out for simple things like tractors, fertilizers, and irrigation canals, just basic stuff. And you know, they'd be outraged that the fashionable elitists back home were trying to deny them these things. So I think there's an interesting environmental point to be made there, that when you're starving, you just need the basics. And this issue of climate change is a huge issue, of course. It can't be underplayed. So this, again, this is a little dated, but this is just a map. I'm sure there's heat waves that have exceeded this heat wave actually from 2012, showing this exceptional drought levels throughout these wheat producing areas of the United States. I just happen to have the data for the United States. There's nothing particular about the United States here. But these droughts, exceptional droughts, and this drought led to huge spikes in food prices globally. FAO is Food and Agriculture Organization. They maintain price indexes globally. Huge spikes, and these spikes led to wars in some areas of the world. You know, this terrible problem of food security. And this is an example of intensive farming, over-intensive farming. This was, again, in the US. This was where the land was stripped, essentially. The Dust Bowl was created in the 30s. It was a period of severe dust storms that greatly damaged the ecology and agriculture of the US and Canada. It was to do with farming methods. They failed to adopt farming methods to prevent wind erosion. And it was just this simple factor of wind erosion that caused this total change of the landscape. And so this is why we talk about sustainable intensification. We need to grow more crops with less, but in a sustainable way, not an unsustainable way. And things just seem to get worse and worse. So there's increasing global hunger. It's a global trend. There's less and less land available for agriculture as cities develop and we urbanize. And interestingly, global improvements in yield, crop yields, are steadily reducing. Now, these are improvements. These are like a 1% improvement in yield, 2% improvement in yield. But this is a rolling 10-year average growth in crop yields. So corn, soybean, rice, and wheat. You can see these improvements in yields are steadily declining. So this somewhat whimsically, this is the conclusion you would draw is that we're all doomed. And this isn't untrue, I'm afraid. I'm sorry to remind you. I'm afraid you are going to die. Unless we can solve aging very quickly, we are doomed. But what can we do about this? What can we do about these problems, these seemingly insurmountable problems? Well, we need a second green revolution. This is really where I want to convince you that through genetics and genomics, probably you don't need convincing. We can create a gene revolution. And this is really being driven by the reduced cost of genome sequencing. I'm sure, as you know well, next generation sequencing is dramatically reducing the cost of genome sequencing. So back in 2002, the rice genome was envisioned as the serial genome. So the rice genome would be the tool that we use to look at wheat, maize, soy. It was sorghum. It was the genome that would represent all cereals. Whereas now, we have whole genome sequencing of essentially every major wheat variety. This was back a few years ago when I was writing these slides. I know this project is well underway now. We have genome sequences essentially of every major wheat variety, which is tremendous innovation. So that's a huge development. I'll talk about how we can start translating research using these new genomic resources. And of course, we have ever-improving algorithms and databases for genotype to phenotype association. So looking at the genome and understanding which genes are responsible for which traits. And then that information feeds through to marker-assisted breeding. Marker-assisted breeding is a very important concept. And it's not genetic engineering. It's about sophisticated methods of breeding using all the available technology and data. It's a data-driven breeding program. And these algorithms like genomic selection, and they lead to optimized breeding programs. Of course, we can think about tools for example, CRISPR to re-engineer plant genomes. But currently in Europe, for example, the use of CRISPR is classified as genetically modified, and there are all sorts of issues around the use of CRISPR. So I'll give you a couple of examples now about how genomic insights translate into improved food security. You talk about rice and talk about wheat. So this is an example from IRI, which is the International Rice Research Institute with large centers in India and Indonesia, I think. Yes, the Center for Rice Research is the headquarters in Indonesia. So if you didn't know, rice dies within a couple of days of submergence. If rice gets flooded, it will die after a couple of days. So it's estimated across Asia, there's 20 million hectares of rice prone to flooding. So it's a huge problem. And India and Bangladesh, there's 5 million hectares of rice are flooded during most seasons. So there are natural varieties of rice that are resistant to resistant to flooding. So those analysis of those natural lines has discovered this gene sub1. And this is looking at an elite line of rice with the sub1 gene there, flooded. The rice field is now flooded. And this is the same elite variety of rice, IR64, on the left with this sub1 gene introgressed. And you see this sub1 gene confers protection to the flooding, tremendous protection. So the IR64 alone starts to catch up by the end of the growing season, but you can see the rice on the left is ripening more effectively. And after harvest, that's the harvest. I'll just pause there, yield comparison. Oops. So the sub1 gene has allowed 3.8 tons per hectare. The wild type rice, or the elite variety, actually, it's not exactly wild type, but the elite variety alone, 1.4 tons per hectare. So this was a process of, it's a tremendous amount of work that's gone on at IRI. The marker-assisted breeding introgressed the gene into high-yielding Asian and Indian varieties. So introgression means bringing in the sub1 gene into these high-yielding varieties. And just this small thing, you can see the difference in yield almost, or is it more than double the yield? Let's see. 3.8, 1.4, yeah, nearly double the yield. More than double the yield, sorry. So these are the kind of things that we can start to do based on analyzing the wild populations of rice. We can find these genes of interest. And as I said, these are marker-assisted breeding is used to introgress these genes. So this is an example of a very sophisticated breeding program. You don't have to look at the details here, but just to give you an example of the sophistication, MAS here stands for marker-assisted breeding. So you're starting with a commercial, high-yielding variety, and you're bringing in this wild type that has a gene of interest. The idea is that we want to combine these different genes of interest from these different wild types to make one commercial variety that contains all of these properties. I'll talk about some of the things, some of the desirable characteristics you might want in a second. There's an F1 cross to produce this first generation of offspring. Marker-assisted breeding is used to help select these F1. Now these, although they're crossed with a high-yielding variety, they won't be high-yielding. They'll bring in, the genomic drag brings in a variety of genes. 3 rounds of this breeding, selfing back to the high-yielding variety, using marker-assisted breeding at each step. Then we cross these different lines, and then these different lines are crossed, and then there's more marker-assisted breeding, more back crossing, more marker-assisted breeding, more back crossing, and then selfing to finally produce. So these programs are very sophisticated, and it can take a long time. So you can imagine without marker-assisted breeding, you would have to grow this variety up. You would have to test this variety for the trait of interest. You'd have to carefully select. Each one of these stages is now done in the lab at the seed level. We can see the markers in the genome that we want, and we know they're associated with the genes of interest. We can just use lab-based techniques to select seeds for the next round, for fertilizing the next round, and then we can use prickle pollination and other techniques, other molecular biology techniques to produce the next generation of seeds from germplasm. We don't have to grow the plant up, reach maturity, test it in the field. This massively accelerates the process of developing new varieties from literally decades. It can literally take decades to produce, for example, a new variety of potatoes. These techniques can bring this down below 10 years and eventually even faster. This is an example in wheat. It was a very important crop as well, of course, in addition to rice. This is just a very simple study showing a field of wheat. Each of these different plots is a different variety of wheat, a different genotype, a different product of one of these crosses we saw. Each of these plots is grown up sort of independently of the other. And what they're looking at here is the number of aphids per grams per leaf on the wheat leaves. See, on average, most of the wheat has like a similar amount of aphids in grams per leaf. But you can see there's variability across these wild varieties. So that some wild varieties even have a lot of aphids in grams per leaf. But this variety here, for whatever reason, whatever biology, it has much less aphids grams per leaf than all of these others in this field. So once you've identified these varieties, you can develop qualitative trait loci, and you can drill down to the genes, and you can develop markers for those genes, and those markers can feed into breeding programs, and then we can engineer or breed, I should say, varieties of wheat which are much less prone to aphid infection, and that will reduce in less pesticide use, which means we can get more from less. So we're producing more food with less inputs by using this incredible natural variety that exists, that has essentially been lost due to domestication. So this variety of biology, essentially, that wheat exhibits, or grasses exhibit, was lost as wheat was brought into the food chain and turned into high-yielding cultivars. So just to sort of reiterate this point, the large-scale phenotyping of wheat in this example directly leads to the development of qualitative trait loci, QTLs. The QTLs allow you to identify the causative genes or markers, and then we use marker-assisted breeding to bring those traits of interest into the high-yielding commercial varieties. So some of the things we might want to improve are the improved responses to abiotic stress, so drought resistance, as we saw. Improved response to biotic stress, that's disease resistance, so think of nematodes, blight, and so on. Improved processing characteristics, so that the farmer can, for example, harvest the wheat, the whole field is ready at the same time. So one harvest, all the wheat is ready at the same time. That's consistency. They all grow to the same height, and so on. And improved fertilizer use, for example, to do more with less. And there's a huge amount of stuff that we can select from in order to do this. So one thing that just came to mind, for example, is looking at Brassica. I did some work with a Brassica research group, and they were looking at pollen production in Brassicas. So in the UK, we often grow rapeseed oil in rotation with wheat, because Brassicas are legumes. They bring nitrogen into the soil, which is useful for the wheat growth in the next season. When they looked at the varieties, across the varieties of Brassicas, they saw that some Brassicas were producing a lot of pollen, so they were very beneficial to the pollinators. And some varieties were producing essentially no pollen whatsoever. And some of the commercial varieties that were being grown widely essentially had no pollen whatsoever. So by improving the crop's relationship with the ecosystem, with pollinators, we can do more with less and make more use of these ecosystem services, such as the calcium and the nitrogen that's available in the soil. Some plants dig deeper roots and can more effectively work with the microrhizomes, the fungal interactions, to better utilize the resources in the soil, because a lot of nitrogen fertilizer is just washed off. So if we can develop plants with better nitrogen use efficiency, it's hugely beneficial to the environment. So moving on to what we do in Ensemble plants, there's a variety of things that this is actually from the EMBL. They had a big project called Elixir, where they want to build, and this is all open, free, and available to the world. They're building these platforms to transfer data, storage of data, and computing over the data. I would really recommend you look at the EMBL resources and look at the databases available to everyone, so that you can actually use them to store your own data. If you're producing a lot of data, submit it to the archives. You can keep it under embargo, and you can use their data storage and computing and transfer facilities. They're using this base layer to build these pillars, so-called, especially around standards. It's extremely important to have data in standard formats. They're developing tools that can work on these standards, and they're providing technical services, so support to people who want to use these tools and so on. That includes training, for example. So EMBL has and EBI has a huge number of training courses. Some of these are online training courses. So if you write a cover letter and send your CV, you can attend some of these courses to do with next generation sequencing analysis, how to use the archives, how to search the archives, and so on. I really recommend, again, making use of those resources that are available completely free. And these are a variety of sort of data-intensive sciences that are supported by these pillars. You see, training is a big part of that. So Ensemble plants, we sort of take this infrastructure and we build on the Ensemble genomes. So Ensemble genomes is a very useful framework for reference genomes, variation data, and comparative analysis. So whilst I was group leader of Ensemble plants, I was using the Ensemble technology to make use of their facilities for reference genomes, variation, and comparative analysis. And these are the kind of things we were doing at Ensemble plants, like marker selection, functional inference, and so on. I'll try and get to that briefly to show you how you can do some of these analyses. For example, based on work in sorghum, you can translate that into work in barley, wheat, maize, rice, or work in rice. You can take that into other crops. So this is where we get into Ensemble plants. I've used quite a lot of my time, so I just say we have data for genomes, we have comparative analyses, we have variation data, and regulation data is starting to come. I just want to ask the audience, who's particularly interested in genome data, comparative data, variation data, or regulation data? And I can just cover that topic instead of going through the whole slides. Yeah. That's me.
Yeah. Yeah, actually, I have one question. Can I ask now or after presentation?
Yes, please, now.
Okay, thank you so much for providing the information. This revolutionary database, actually, we have utilized this frequently in our so many papers aside this, actually, for the identification of gene and transcription factor families in wheat. So I need one information, actually. Is there how many wheat varieties have been sequenced yet, actually? And what is your plan to add these data for ensemble plants? Because as per my best of knowledge, there are 14 cultivars for wheat, actually, available on ensemble plants. So how many entry would you like to add on ensemble actually in future? And is there second thing is there any plan to about the to add the expression data actually
To have
add
the expression data? Yes,
yes.
yes.
So these are
So
the two questions actually.
yeah, I don't have slides here for the expression data, but part of what I did at Ensemble Plants was I developed a pipeline to look at the Array Express, you know, the database for holding expression data. So the Gene Expression Atlas and Array Express are just, you know, they were 10 meters away from the Ensemble Plants office. So we developed an automatic pipeline for pulling essentially every month all the data, plant-related data from Array Express, aligning it to the genomes and then presenting it in the browser. So this is something which was being done routinely. In terms of the varieties, I'll just check this comparative analysis. So as you know, wheat is a polyploid, and plants seem to have many instances of this polyploidization. This is the history of wheat. So I'm just skipping forward to these. These are the gene trees we show, which show currently we have the D genome, the B genome, and the A genome of wheat separately with these ancestral progenitor wheat species, Aegyptiaceae, Triticum maratu, and then Hordeum vulgari, of course, barley as an outgroup, and here's Brassica. And then all the alignments of these genes are there. So what you're talking about is when are we going to add the cultivars into this gene tree so that we can start looking at the genes specifically of these cultivars? It's a very good question. So I've got some slides from my colleague who's the current Ensemble Plants team leader. He sent me these slides as an update, a very recent update. So what we're working on now is essentially the concept of the pangenome. So we can continue adding varieties ad infinitum. You know, essentially there's as many varieties as you want, because the question is, how do we handle that data? How do we make that data useful? And that's where the concept of the pangenome comes in. So you have different cultivars with different core genes, different dispensable genes that they have in common with other cultivars, and different unique genes. And this is the sort of data set that we want to provide. And it's coming in a variety of forms from wheat and rice, the sorghum there, and there's lots of other species in Ensemble Plants. So the person to contact now is Guy Namathi. So Guy, I can put his contact details in the chat. He's the current Ensemble Plants team leader, and he's pushing this initiative for pangenomes. It's just showing how popular wheat is. Wheat is now, Ensemble was started with the human genome as the main driver, but now wheat is starting to rival the human genome in terms of accesses. So the pangenome, we just talked through the definition there. The pangenome is a huge concept. I'm sure you're aware of it. So we're working, or Guy is now working with the Wheat Pangenome Initiative. We're getting funding, Ensemble Plants is getting funding from the Wheat Pangenome Initiative. I'm trying to see if he's talking about, the cultivar view in Ensemble Plants. So let's see what we have here. Here we have it. This is what you're after, I think. So thank you for asking, because I would never have thought to just go and present these slides necessarily. So here's stretching message from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10,
14
11,
actually.
12, 13, 14. Right. So those are the 14 that you mentioned, yeah. So this framework will allow us now to expand the range of cultivars that are in Ensemble plants. And I actually don't know, because I've not been involved recently, what the plans are. I don't know what the plans are to expand that in collaboration with 10K Genome Project, because I was trying to sequence 10,000 wheat genomes. So if you talk to Guy, he will have clear timelines about exactly when these genomes are going to be added into Ensemble plants. Yeah, sorry, I don't know actually. I wondered if it would be on these slides, but it isn't. Yep. Yes, that's right. So we were trying to work, for example, with Erie in the Rice 3000 Genomes project to bring all that data in. One way in which we can do that is through variation. So although the pan-genome concept is being pushed and developed, where are we? This is the core. That's of course out of date now. Variation. So by pulling in this variation data, we can show data from, for example, the Rice 3000 Genomes project. So, and of course, there was a big push for Brassica, which I know is very important crop for India in particular. Where's the Brassicas? Yeah, there we go. The BMAP, 150 Brassicas are being sequenced and pulled in to Ensemble plants. So through the variation interface, we can see the particular alleles or sequences unique to a particular variety without having the whole genome imported. So it's sort of like a shortcut to whole genome sequencing is just to aggregate this variation data. Yeah, and I agree, there's a huge amount of germplasm out there. So hopefully, I mean, there's two issues here. One is just coordination, bringing the data together under a standard framework like Ensemble plants or any other, standard-based open source data infrastructure. I think that's critical. But there's another concept, which is who owns the data. And I know that's a big issue. Some of these wheat varieties, some of these rice varieties, brassica varieties are essentially natural resources of India. They have emerged in the Indian ecosystem, and they are unique to India, for example. And it's part of your natural resource. If it was oil, there's a very clear pathway to revenue. But when it's information, and that information can be just tapped into and extracted and then shared, these principles of open source, global freedom, everyone should have access to the data. Well, actually, who receives the benefit? Should that benefit just go to big agriculture, big players who can develop new varieties and sell those varieties back? But actually, this is our variety, it grew here. So I think there's some interesting work to be done on data provenance and this sort of chain of provenance that exists with this biological data. It's an interesting concept for me. Always having been an open source proponent, open source advocate, well, actually, this data is a bit different. These varieties are essentially natural resources. I have too many slides, unfortunately. So let's see, I haven't talked about the regulation data here, unfortunately. So this is just showing the, the importance of regulatory data in Arabidopsis. I was amazed by this paper when I saw this paper. All these Arabidopsis have the same identical genome, and these are stably inherited methylation markers, so such that you can produce a gene tree, not a gene tree, of course, a phylogenetic tree purely based on these, well, I want to call it a genetic map, but it's an epigenetic map. You can build an epigenetic map of the chromosome using these epigenetic markers across the identical genome, just showing the stability and importance of these epigenetic markers. These two plants have the same genome but different epigenetic maps. And that kind of data is slowly feeding into through the regulation interfaces that we get from Ensemble. We're starting to see that data come up. So yeah, I guess I'll leave it there and ask for questions. This certainly hasn't been all my work, a huge number of people involved. And as I said, I'll put Guy's email in the chat now so that anyone can email him if they want information about the latest release of Ensemble plants. Of course. Yep. Mm. No, it's a very good point. I think one of the bottlenecks, sorry, just to mention, I thought I was sharing my screen during the last part of my talk, so it's probably a bit confusing for you. Did you see the slides from Guy, or was I just talking? Okay, sorry. This was the 14 I was counting, and yeah, sorry. So, and this was the team during my time. So one of the difficulties whilst we were developing the wheat exome capture chip is literally that. It's the exome capture chip is always a trade-off. You want to get the chip developed as quickly as possible, so you want to have this chip ready and commercially available, but you also want to have the chip capture all of the exomes from all of the varieties. So it's sort of a chicken and egg problem, that you want to collect the varieties' whole genomes in order to study the exomes, in order to make the best exome chip possible to go out and look at more varieties. So it's, if you miss an exome off the exome chip, you will never see that gene in your exome capture. So it took a long time for people to say, right, this is version 1 of the exome capture chip in wheat. But as you say, it's then reduces the cost of genome sequencing, because you're just sequencing that small fraction of the genome which is relevant for biology as far as we know. Because wheat, I could actually borrow some of Guy's slides now. Wheat is a huge genome. If you didn't, I'm sure anyone working on wheat knows this problem. But it's a 17 gigabase genome compared to human of 3 gigabase, or rice down here. So most of that, of course, is repeat sequences. So why sequence 70 gigabase when you can probably just sequence this little portion that's common to rice, the important genes? So the exome capture is, developing the exome capture chips are part of the puzzle. It takes some time and it takes some cost. And of course, all the time genome sequencing is improving. So the latest data from Oxford Nanopore, the latest techniques from BGI, the nanopore, excuse me, the DNA ball-based sequencing. So all the time it's getting cheaper and cheaper to sequence genome data, such that when I was involved with grants where they said, okay, we'll spend the first fraction of the grant on sequencing now, but we'll save a fraction of the grant for sequencing in 3 years, because in 3 years' time, it will be so much cheaper to sequence. So it's just, do you spend a year developing an exome capture chip, or would you rather just sequence whole genomes? I think as same goes for the SNP chips for variation, there's a huge potential there just to very cheaply probe the variation across a gene bank. You look at all the germplasms and probe the variation very cheaply using a SNP chip, but it's not the whole picture. And would you actually do better to just wait a year or two and then sequence the whole genomes? Yeah, I think that's one of the reasons. Yeah.
Can you see me?